Yeah, thank you. Thanks for the opportunity to present here. So far, we've heard a lot about like deforestation and the land use changes after deforestation. But today I want to shift the focus a little bit to restoration and especially more specific reforestation efforts globally. In the first part, I will quickly reiterate what we did for one of the monitors in the project, the World Reforestation Monitor, and the outputs we produced there. And in the second part, which is the emphasis of this presentation, I will talk about how we use all these products that were developed in the Open Earth Monitor community to really start to track restoration efforts globally and how we can sort of estimate an impact that this does. Okay, let's quickly reiterate the World Reforestation Monitor. And the idea is that we map certain biophysical variables of vegetation, including, for instance, the leaf area index within a canopy. And we can do this using the help of the so-called mechanistic radiative transfer model. So they simulate, based on all the traits in a canopy, they simulate the expectant reflectance curve, and then we can use this to infer, for instance, variables like leaf area index again. And we have then sort of used in situ on-the-ground data to do both the validation and the calibration of these mechanistic model outputs. This led to development of 3 different variables, the effective leaf area index, the FAPAR, and fractional vegetation cover, which we mapped at annual time intervals up to 20 meters spatial resolution. And these are aggregated to annual products because we think it's most useful for tracking restoration efforts by looking at the peak growing season throughout the year. And the annual products make it end users then easier to analyze the products and not having to deal with the within year variation. So this is still, unfortunately, in preprint state. I hope to have it published sooner, but it will still take a couple of months. But you can look at the data products, the preprint, and a Google Earth Engine app, as well as the Earth Monitor app. Okay, now this was sort of the things we developed for the Open Earth Monitor, but now let's look at restoration and how we can use all the products from OEMC to evaluate global restoration efforts. And this is very important as you're aware, we are in this declared decade of ecosystem restoration. So it's now really a priority to go from these degraded ecosystems back to healthy ecosystems that can support biodiversity, but can also support us humans in the long term. And for tracking these reforestation efforts, we work together with our stakeholder, which is restore.eco. It's a global platform which connects all these restoration efforts, and it tries to deliver transparent, science-based, practical data to the practitioners on the ground. So basically, you can draw a polygon and you get lots of scientific insights based on published maps by the whole community. And importantly, it's also a way of connecting to other restoration projects. Restoration is not as simple as it looks, and you need to know in your area what has been tried, what has been done for you to develop effective strategies. Now, we were given this restore polygon data, and when you work with like this real world user uploaded data, you will have lots of problems, and I will quickly go through some examples and the steps of filtering that was required to get good restoration projects that we can actually analyze the interventions of. So first is like simple checks, like 2 small polygons, 2 big polygons, polygons with like weird shapes. If it has like 3 vertices, then we don't consider them as a reliable project. The second step was we had many overlapping polygons. For instance, here we're in Madagascar and you have lots of small restoration projects. and then one large project boundary around it. So we exclude this sort of larger aggregative meta projects because we really want the local project information and to analyze these. And then here's another example where you have this very small scale overlap between polygons, and we had to do a couple of filtering and pre-processing steps to get the most out of the restore database. So for instance, on the left, you can see two overlapping polygons with different intervention years. And we really want to know when was the intervention carried out, such that we can compare the time series before and after intervention. So for instance, in this case, we had all these small overlaps and one big overlap. So in that case, we keep the earliest intervention year for this polygon and exclude like all the overlapping sections where the overlaps are very small. So you see these overlaps on the right-hand side, they get excluded from the analysis. So this is what we are left with. One other problem that we couldn't really address is when you look at some of these uploaded projects, you have one here in Campeche, Mexico. They give you an intervention year for the whole project, sort of. But if you then dig a little bit deeper and go to the original third-party platform that uploaded this, you see that these intervention efforts are actually much more fine-grained. So the plantings occur at sub-hectare levels, but the whole project polygon they upload can be much, much larger. So we have like this uncertainty in the intervention year, or like a bit of fuzzy intervention year that they gave us. Okay, now to the project that we used for the analysis that remained after all this filtering. We specifically focus on the post-intervention target to be natural forest. So it's not supposed to be production forest or mangroves or grass or savannas and so on. It's really natural forest as a target that they declared. And we look at intervention years starting between 2000 and 2024, because this is what covers most of the products that were developed here in Open Earth Monitor. And then one important consideration is the intervention type. You can think of different types, including active restoration, like really large scale planting of tree species, of native communities. You can think of like natural regeneration, passive or assisted, and conservation efforts, and all the other, which might also include like agroforestry. And they are distributed across the globe, not equally, of course, but most of the observations are from the tropical moist forest biome. Now, let's look at the data that was generated here in the Open Earth Monitor. First, we look at the effective leaf area index that was part of the World Reforestation Monitor. And we can sort of, just by plotting the time series for each of the projects and then standardizing first by intervention year and then by the mean LAI before intervention, we can derive these plots and just make a smoother over it to see whether we expect like an increase overall across the database or not. The same you can do for a bit longer time series. This is the median vegetation height, or SVH, short vegetation height product, which gives you, if you think about the vertical profile of vegetation, where the median of this vertical profile is sort of the mass of the density. And also here we see these slight increases across all the different projects. But now we want to quantify this a little bit. Sorry, it skipped over. So the first way to quantify it is using a linear mixed effects model. You don't see the headers here. Yeah, I don't know how to solve it. Yeah, let me just continue. So we want to develop a model where we have each different project starts at a different baseline. Then we have a trend before intervention, and then how the trend after intervention changes. And sort of these random effects take into account the site level variability. So each site can start at a different baseline, and each site can have different slopes post and pre-intervention. And we're more interested in these across all sites. What are the group level effects, sort of, not the individual projects in this first analysis. And these are the group level effects for the 3 different categories: active, natural, and conservation. So the first thing you see that they start at much different baselines. So this makes it hard to compare the magnitudes of the effects that you get, because you have to know that they start from different baselines. Conservation, of course, has much more vegetation compared to the other categories. And then you can take into effect again the site level variability described by this random effects terms, and predict for each site where it is after 10 years of intervention. And you get these histograms, which, if you would have no change, you would expect the sites to cluster around 0, but they're like clustering above the 0. With, like, if you interpret it, on average, you have around 31 centimeter increase in this median vegetation height in 10 years in the active restoration category. Okay, so this is like a simple before-after analysis, a linear mixed effects models with all its flaws that come with it. For instance, the counterfactual that we assume here is that the mean in the pre-treatment phase would continue, and we assess like the difference to this mean after intervention. Now we want to make it a bit more, sorry. Yeah, seems to sometimes just continue. Sorry about that. So we applied a generalized synthetic controls approach, which means we take all the not yet treated sites, or all the sites that were treated late in our panel, and use them as potential control sites for the earlier treated ones. And when we do this for like a specific ecoregion, here in Brazil, a panel looks like this. So you have treatment sites on top, with the dark blue being the phase where the treatment is in effect, and the light blue is sort of the control phase before, and you have pure control sites, which were those with very late intervention years. And this generalized synthetic control method then allows you to quantify this, this change for each of the site. So in effect, for each site, it sort of constructs a counterfactual based on the pool of control sites that were there. Here, this is for one specific site. You see all the control pool donor sort of in the more transparent lines, and you see the counterfactual, the dashed line, and the effective observed line, the solid line. And the method also assumes like there can be background effects. You can think of like an El Nino in this region that affects all the sites. So it actually estimates these background effects and calculates them and takes them into account to really get to the causal effect. And then you can aggregate this to the full ecoregion level to get like a average treatment effect, for instance, here for the whole ecoregion, and see how this changes. Now, when we do this globally, we take all the sites and apply the same methodology for the globe. We can see that this increase that we saw in the exploratory data analysis also holds here when we let the model sort of construct a counterfactual for each of the treatment sites. Okay, but what are potential pitfalls when doing this analysis? So now we have like this global donor pool of control sites, and maybe it's not so sensible to construct a synthetic control based on this whole global pool of sites. Also, we have like a site selection bias because we only include the restoration sites on restore. So maybe they systematically differ from never treated sound sites. And as mentioned, we have this sort of uncertain intervention timing. So the top two we tried to address with looking at a more localized donor pool. So we build new control sites for each treated site, and then we sort of match this. So this is how it looks in practice. So this would be one of the restoration sites with quite a complex geometry. Then we create a buffer around it of like 10 kilometers for potential pixel locations which have similar characteristics to the ones in the treatment site. And we have some exclusion buffer. And then we match these pixels sampled within the control site using the alpha earth embeddings to find pixels which are very similar to the ones in the treatment sites 1 year before intervention. So we are restricted to interventions now from 2018 later. And then for each site, we sort of create this synthetic control time series, which is a weighted average of all these sort of similar pixel locations to construct a sense of a counterfactual. Yeah, this is basically what the generalized synthetic control now does with this added new time series, which are from these sampled control pixel locations. If you apply this for a country like Brazil, you can then get an average treatment effect across the whole country like this. And you can split this analysis based on different hierarchies. So you can do it on a biome level, on an ecoregion level, or on a global level. And then you get results, treatment effect results for each of these hierarchies. So this would be on the ecoregion level, taking the most abundant ecoregions in our data set. And let's look at some examples. Here, for instance, we take the ecoregion Madagascar humid forests, and we look at like a single site within this ecoregion, and we can then actually get a treatment effect for this specific site because we have constructed a counterfactual for each of our treatment site based on this new donor pool of control time series data. So these are like examples where it works well. So you really see a steep increase. But of course, you can also use the method then to look at sites where it might have worked not so well, or maybe there was like a deforestation or degradation after they claimed that they started the intervention year. Okay, and you can also apply this on a global scale to look at different intervention types, and I'll just show you the results of these graphs sort of on a global scale, where you can see both in active and natural regeneration that we really have this recovery across all sites. And for each of the treatment sites, it implicitly created a counterfactual using this global new pool of donor sites. So this was, to wrap up, we have the second method where we use the not yet treated sites as controls. And in the third step, we sort of created new control sites for each treatment site to enhance our control donor pool a bit more. So in conclusion, we tried to use the products developed within the consortium, and then I now focused on median vegetation height, but the conclusions are similar if we would apply to, for instance, the GPP, the uncalibrated one. And we can really track these small-scale restoration efforts, which are like sub-hectare, by sort of constructing a counterfactual time series for each of the treatment sites. And as an outlook, we will use an updated version of the Restore database and maybe other project data sources in the future to apply this analysis to. And one thing which would be nice is to integrate everything into a more multimodal restoration effect framework and not just focus on single variables. And maybe as a last point on the bottom, what we do in the EU Biodiversity Monitor is more look at the spatial heterogeneity, so thinking about spectral and functional diversity and how this recovers after restoration efforts. So yeah, with that, I'm finished, and I'll thank everyone here, Open Earth Monitor and my lab, and especially my supervisors.
Thank you. Wow, this is really it's a complex work, and I'm very happy we're combining forces. This is really this was what this project was all about, to put and combine forces and do this type of analysis. This is global analysis, right? It can be applied anywhere.
Yeah.
I really like it. It's a bit complex. We need some time to digest, and let's start with some questions, but I'll kickstart because something's bothering me. So like for this median vegetation height, we also have uncertainty, right? You have uncertainty bands. Can you incorporate uncertainty in this type of testing and this linear
I could
mixed
think
model?
of like a bootstrapping approach,
Okay.
where you repeat the analysis with like
Simulated error, yeah.
Yeah.
Okay.
Something like this. We repeat it like 100 times and always sample from the uncertainty.
Yeah, and then if the uncertainty is high, you'll probably get a less effect, and if the uncertainty is less, then it's more crisp.
Yeah.
Okay. Questions, please. Robert, Greg, and Stefan. Robert, you start. Microphone coming.
Yeah, thank you for the nice presentation. So during the introduction, you said you only did this in natural forests, right? We excluded mangrove and savanna. I was just curious on the definition of a natural forest. Like which definition did you use, and why exclude mangrove? Because I believe the restoration efforts which are also done in mangrove and savanna, I think.
Yeah. Here I wanted to have like a comparable set of projects, and this is all self-reported data. So they have like a category of 20 different post-intervention land use land cover types that they can choose from. Let's say grassland, grazing, production forest, natural forest, and so on. And we wanted to really focus on a comparable set of projects where the aim is to restore a natural forest. So this is self-reported data from each project that they say we want to reestablish a natural forest.
But mangrove is also a natural forest, right?
Yeah. I will plan to repeat the analysis, but then just for mangrove sites, sort of where they want to restore a mangrove. So the ideas would be to repeat it for these different natural ecosystems.
Next question, Greg, please.
Thanks, Felix. Nice to see the progress. One thing though, I wonder, I mean, if you're, from what I understand, I don't quite see if you can you show the added value of using this on a product like the medium height or the GPP that you say, or even your LAI, because from my understanding, for what you do now, you're trying to see increase in biomass or greenness or whatever. So in the end, it would have been more I mean, I would think first thing would be to do it on, I don't know, NDVI, the simplest thing that has no assumptions.
Yeah,
I guess
I've done the same analysis with NDVI, and it gives quite similar results.
So can you say something about the added value of doing it on these more sophisticated products? I mean, do we gain something on it, or do we just see that there's more greenness? Because I think the greenness is good, but then why would we need LAI and all these things for this?
Yeah, I think one thing is like if you then also do it for, like we use the NDVI from Landsat from your group, but if you repeat it with like different, like short vegetation height or the LAI, which is based from Sentinel, it just gives a bit more confidence in the signal because you have different independent sensors giving you the same result. But I agree, in the end, if the conclusions are the same for short vegetation height and NDVI, you can do it with both. I like the interpretation more of the median vegetation height compared to the NDVI.
But it depends on the quality. I mean, yes, I hope
But
it's
just
good.
a second, but look, so you can have some country that the whole country gets, let's say it's a wetter years or something, and everything goes greener.
Yeah.
I think what I understand it, this is a very robust way to say, well, in comparison to other area, how much it changes in comparison to that. So it's not just a slope. He plots it
Sure,
as
sure,
the
sure.
slope.
Exactly. No,
But
sure.
but you can now really test it whether it has, is it really different? Because it could be that the whole country gets a bit greener.
No, for sure. But this
You know.
applies to whether you do it on one index or one other thing. So that's great that you localize. So I think that's good. I wonder if you could show a bit more because you've done it for different things, whether you can actually say, I mean, one signal might be stronger on like that you reach something before or after. You see what I mean?
Yeah.
So that you can add value to the fact that you applied on other things. Because otherwise, I would say then it's more prudent to do something on a less interpreted. I mean, every time we try to get a product that is more complex, you have assumptions that go with it. And it's nice to give it a name like LAI, but it's not the LAI of each leaf. And it's like the signal from the satellite more raw can be actually as good and more reliable in a way. No, but so if you can show that what you did, you see the signal go up or down. I mean, that would be interesting to evaluate that. I think.
Yeah, and sort of take into account the correlation between like the NDVI and the median vegetation height for a given site, and really look at when these signals diverge, sort of when you have an NDVI going down but a SVH going up, sort of. Yeah.
Here's one more question from Stefan, please.
Yeah, just maybe also some observations and comments. So it's going a bit in a similar direction. So I'm just wondering because, I mean, I think there is an advantage, of course, of using those global products, but as we know, these global products do have quite some uncertainty. What you don't want is to have this uncertainty necessarily too much in your site. So what could be done also is you could consider your sites as a separate kind of area of looking at things. So for example, you collect, I mean, this would be an additional exercise, but to make sure you're reducing your uncertainty in these sites by collecting, for example, more training data in those sites, and then rerunning the thing just because you only need data on the sites, right? That's what you're looking at. So you could really do a lot in reducing this uncertainty which you have in the global products by more training data, but you could potentially also, since there are people on the ground, I guess, in these places, maybe get them to collect some data, some validation data, and then you could also more refine this, right? So you have different treatments. They as you have shown, they are very, very different. So some of them, they will have as an objective to increase biomass. Some others, they do some restoration, but they will not probably increase biomass. So you could try to separate this out and say, I have X, Y, Z trying to increase biomass. The others ones, they are trying to increase some other parameters. So you could really specifically target what you're looking at on what they are promising in the project to do and really tease that out more. So I think there are endless things you could extend this, but it's a good starting point. So it's interesting. Yeah, thanks.
Yeah, it's a great work, fantastic. It's very impressive, and continue, please. And also, this is the restoration projects, but you can convert it the same method you could apply for degradation, right? So you go out of the restore.eco and you can do the same analysis where they do deforestation or whatever, and you can say you have a negative trend here, right? So it's a same method. I mean, nothing really changes, right? It's just the plots will show more like downhill.
Yeah.
So yeah, no, fantastic. Thank you so much, Felix, and